Okay, so hello everyone. Today we're gonna discuss about PEROXISOME. So the PEROXISOME was discovered by Kristen Deloye. This was discovered by Kristen Deloye. Fine. So PEROXISOME is basically the micro body. What? This is micro body. Now you be thinking that what actually micro body is. So micro body is when small, we can say, when single membrane bounded organelles like minute-like vesicles combine together. So those are known as microbodies. Let me write here. When single What is that? When single membrane bound organelle like minute vesicles called microbody. जब सिंगल मेम्ब्रेन बाउंडेड ऑर्गेनल बाउंड होता है और एक वेसिकल बनाता है, so that is known as microbody. Okay, so now this peroxisome and glycosome both are what? Microbodies. Glycosome will be discussed later on. So now microbodies are basically very small, which we can't see from a naked eye. Very small. We have to use the electron microscope to see these microbodies. Fine. A peroxisome is found in both plants as well as in animals. Found in plants and animals. Now peroxisome is found in both, but the main function or main role peroxisome play in the animals. So hence this is also known as animal body, microbody, because it play main role in animals. Fine. Hopefully it is clear to you. Now here the peroxisome is found in liver, kidney and brain cells. Liver, kidney and brain cells. The major function of peroxisome is to the formation and decomposition of H2O2. What? Formation and decomposition of H2O2. This is what? Hydrogen peroxide. This is very toxic. Okay, hydrogen peroxide. Now peroxide contain different enzymes. Those are peroxidase, catalase and glycolic oxidase. May I write here enzymes. So those are basically which peroxidase, catalase. Third we have glycolic oxidase. Fine, these are the three enzymes which help peroxisome to convert different pieces in the breakdown or formation. Fine. Peroxisome in plants, it is involved in photorespiration. In plants, involved in photorespiration. Fine. Now photorespiration is basically the wasteful process, you can say. This we will discuss later on, photorespiration process. Now peroxisome is involved in two things. Number one, we have lipid oxidation. And second, we have amino acid oxidation. Amino acid oxidation. Fine. So here and now let me tell you about. So now first of all we're gonna see the photorespiration, as I wrote the word here, photorespiration. So let's see. So here photorespiration always occurs in C3 plants. Fine, C3 plants. Here what, as I told you here, the formation of H2O2. So this formation is basically oxygen plus H2O and we get H2O2, hydrogen peroxide. Now this is very much toxic. And here let me tell you, photorespiration is process which to be done in the presence of light. To be done in the presence of light. Fine. So here now we are putting the light. Let me draw the picture, then I'll explain you. Okay, so let me explain you this whole diagram. So when oxygen is added with RuBP, ribobiphosphate, when oxygen is added with ribobiphosphate by the help of enzyme, that enzyme is called as Rubisco. Fine. Now oxygen is added by the help of Rubisco. Now this oxygen will be converted into, when we add it by the enzyme, after that this will be converted into Pg3. And now Pg is basically the phosphoglycolate. Fine. Phosphoglycolate. Okay. Phosphoglycolate, then it will be again converted into PGA, phosphoglycerate acid. Fine. When it is converted into phosphoglycerate acid, then it will again go through the same position. So here from this, the CO2 is released and ATP and NADPH is stored. Fine. So this process is known as photorespiration. But if I do talk about this one side, so that is when CO2 will combine with Rubisco biphosphate by the help of enzyme, that enzyme known as Rubisco. Fine. Then it will be converted into what? PGA, phosphoglycerate acid, then it will again convert into PGA to PGA. After that, same Rubisco enzyme. So from here the sugar is released out and ATP and NDPH is released out. Fine. So here I'm going to write somehow process so it will be damn clear to you guys. So this process is also known as photosynthetic. Fine. Now let me tell you a bit different between photorespiration and photosynthesis. So in photosynthesis, basically CO2 intake, while here in the photorespiration O2 intake. Means in the photorespiration we were taking CO2 and combining the Rubisco biphosphate with the help of enzyme Rubisco. In the photorespiration we will be taking oxygen combined with Rubisco biphosphate and by the help of enzyme Rubisco. Fine. Here O2 is removed and here CO2 removed. Here sugar is released. Here energy is stored. Fine. Hopefully till that it's clear to you guys. Now let me tell you one thing is that this is PEROXISOME. Here in the peroxisome we have one core in which things are present. In this core enzymes are present. Enzymes present. So when enzymes are present, so enzymes will make it crystal. Okay, so this is known as crystal core. Hopefully this PEROXISOME is clear to you guys. Bye bye.